Thanks for joining us today. This is our first video in our Ask a Scientist series, and we're really excited to be here today. My name is Dr. Sam Rosolina. I am Vice President of Applied Innovation.
Hi, I'm Dora Taggart. I'm the CEO here at Microbial Insights. It's really exciting to be doing this series for you, and we're looking forward to kind of getting more questions and getting into a diversity of topics this year. So Sam and I will be doing many of these for you. We hope you'll join us on those. And our first topic, Sam, is about petroleum hydrocarbons, and something we love to talk about. So let me go ahead and present the question, and then we can discuss it. So the question that was asked is, how much more is there to learn about microbial degradation processes for common groundwater contaminants like BTEX, MTBE, and naphthalene? And then second part is, how quickly does Microbial Insights incorporate new research into our analysis? So, when I was thinking about this, Sam, I was thinking about the tremendous strides that have been done in this field, right? We've talked about petroleum hydrocarbons since the 70s and 80s, and there's great research that's been done. So we know a lot of these pathways, and it started, right, because we're doing plate counts on the aerobic side. So we started learning about aerobic pathways for degradation, and there's such a mix at these sites with small compounds or PAHs or alkanes even, right? And so researchers were trying to understand these aerobic pathways, and they quickly identified a lot. So today, as you know, we look at around 14 different assays for aerobic degradation of hydrocarbons. And then in the early 2000s, researchers like Dr. Harry Beller and Dr. Kate Scow started figuring out these compounds actually degrade anaerobically. And since these sites become anaerobic very quickly, that was huge. And so they're not recalcitrant. We can actually degrade a lot of these compounds through genes like benzyl succinate synthase or benzene carboxylase. And so they did all this great research, and then we started working with them to make these commercially available. And now we have 6 anaerobic assays that we do on a regular basis. So that gives us 20 different pathways that we can look at. So do we understand petroleum hydrocarbon degradation?
Pretty well, yeah.
Not too bad, right?
We understand it pretty well. And it's interesting because we kind of think about degradation as compounds breaking apart or getting smaller, but a lot of these first steps are actually adding something to the compound. So you mentioned carboxylase, where we're adding a carboxyl group, or benzyl succinate synthase, where we're adding fumarate to a toluene, for example. And you mentioned some of the aerobic compounds or some of the aerobic pathways, and a lot of those are hydroxylation, where we're adding OH groups to these compounds, and that makes it more susceptible to breakdown, and then that can kind of follow that pathway. But there's still a lot we don't know, right? I think you can agree that we have a couple things for benzene. For example, you mentioned the benzyl carboxylase enzyme. There's still a lot that we don't know. We know we can add carboxylase, we can do methylation. There are a lot of genes in those pathways that researchers are still looking into. Yep. And this is not for lack of trying, I would say, right? There's a lot of researchers who have really poured much of their career into this, that they've really been working hard to make this happen.
Very true.
And they're just starting to kind of understand and make some headway on that, but there's still quite a bit that we don't understand. And so what that means for us at Microbial Insights, because as a lot of our clients and colleagues understand, we're on kind of the forefront of innovation, is that we really need to make sure that we're keeping a close eye on academia and seeing what's coming out of these labs. Yeah. But also we want to make sure that we're not jumping the gun too fast. We want to be confident in these studies and have strong validation and repetition in some of these results before we develop these qPCR assays. So we're really looking forward to that happening when we understand those pathways. And I would say one of my favorite things about molecular biology as a whole is that when we're looking at DNA, we can go back to some of these samples, maybe years or even decades later, and we can look for these new gene targets in past samples. So if we do end up coming up with a qPCR target for anaerobic benzene degradation that's new and is important to the industry, you can call us up and say, hey, let's look at this sample from 2023 and see if that target is present. And that may answer a lot of questions about some of the historical nature of your site.
Yep, you can never get that first point back, right? So
That's
preserving that point, we can go back to it as we learn more and more information. And you know, when Sam and I were thinking about this question and talking, one of the things we wanted to make sure we cover with you guys today is there's a myth in our industry that petroleum degraders are ubiquitous. They're present everywhere in the environment. So why do we bother to analyze them? Just don't worry about it, petroleum will go away. Well, we have data to better understand that. So as many of you know, Microbial Insights has been doing this type of analysis with qPCR for over 25 years. And so we have a database of over 250,000 samples from sites all over the world. So what we can do is go in and look at these genes and the detection frequencies and understand better if we can answer that question. So we looked into that, and what we found is for some genes, they are pretty prevalent. They're there about 80% of the time in being detected. But here's the kicker. What's really important for you guys when you're doing site assessment is, even if it's there, is it enough to do the degradation at a rate that is going to make a measurable difference for you? And what we found for this gene that's detected 80% of the time is it's detected at concentrations over 5 orders of magnitude. So if you have 10 cells or 1 million cells, that's going to make a big difference in your degradation. So what we would say is it's still important to analyze even if it can be detected, because you need to see if you have enough to do what you want. And also, other genes are only detected 20% of the time. So they may not be common in the environment. And for anaerobic pathways, you got a 40 to 50% chance of detection. So understanding that, understanding the abundance of the gene, what is present that gives you the genetic potential for that degradation,
Right.
is really critical for this. And so we want to make sure people take that into account. And another question we get a lot, which is about bioaugmentation. For petroleum hydrocarbons, do we need to add the culture in? And so what we think is that, you know, it's probably not necessary to do a bioaugmentation. You just got to get the conditions right. And you know, as Sam and I talk about all the time, when we have these spills, you get a lot of electron donor that the organisms can use as a food source for these petroleum hydrocarbons. But if you don't have enough things for them to breathe, like oxygen or sulfates for them to be able to utilize, those electron acceptors, you're not going to get degradation. They can't do their job. And if the nutrients aren't there to keep them healthy, so just like us, they need the right things to be able to live and thrive and do well in their communities. So we want to encourage you to look at all of those parameters and take that all into context with your microbial assessment so that you can really understand what's happening at those sites.
Yeah, that's great. Absolutely. And one thing that I would say is we, you know, kind of going back to the question of what more do we have to discover about degradation of petroleum hydrocarbons. Another thing that I love about molecular biology is that we have lots of data that we can leverage. So even if we're not discovering a new enzyme or a new organism or a new pathway, we have all of this data, and Dora mentioned our qPCR database and how many thousands of samples are in there. We can leverage this information to better understand how certain data can give us information about a site and even sometimes be predictive about that. And one recent example that we have was a study that we were very excited to be a part of. It goes back to that benzyl succinate synthase gene that Dora mentioned that is so important to the degradation of toluene and ethylbenzene and xylenes in anaerobic conditions. And the goal was to look at this BSSA gene and to be able to estimate first order degradation rate constants for toluene. And so we were able to do that. Really cool. So now we're actually taking all of this data, we're making it even more actionable. And I think this is an important aspect of research and development that people don't often think about, which is that we don't have to discover something new. We can take something that we already have and leverage it for something more. And so I would recommend, there's going to be a QR code on the screen, but I recommend that you look into this paper that we recently published. And also we'll soon be having a qPCR or a BSSA calculator on our website that will help you predict what those rate constants are for your site by inputting toluene concentrations as well as BSSA abundance numbers. And then the last thing I'll say about that is we also have an upcoming webinar from Dr. Giovanni Poloni. And so we'll have a link up for that as well. And that's on February 24th. Now, if you're watching this after February 24th, 2026, I would say to look at our website. We do archive all of our webinars.
Yep, check out that recording.
Well. Yeah.
And we'll put the link on the screen for you for that too.
Yeah, that's right. So I would say overall, as you said, we know a lot about degradation of petroleum hydrocarbons. And especially when you think about the fact that in the mid-1990s, we didn't even know as an industry if benzene could degrade anaerobically at all. And so we're really making huge strides, but there is still quite a bit to learn, I would say. And so we are very grateful for our friends and colleagues in academia so that we can work closely with them. And what that means, again, for us is that they pass that baton on to us and Microbial Insights so that we can develop these qPCR targets and turn them into something more actionable and precision remediation focused rather than kind of a research project. So that's where we fit into this puzzle.
Yeah, it's exciting times. And like Sam said, we still can learn so much more from the data. So we would encourage you to reach out, use your data, reach out to us about questions about the data. And as a community, let's figure out what this all means together, because we do have 25 years of information that gives us something to be more predictive for the future. One of the things that I do want to close out with is we have 2 publications that if you're interested in this topic, I think would be very useful for you. The first is a book chapter that I did with Dr. Trent Key with ExxonMobil, and I'll put the QR code up so that you can download that. That book chapter is available to you. And then we also have a publication that Dr. Kate Clark and I did on 20 years of petroleum hydrocarbons. So you can look at what we summarize and a lot of this information, see, a peer-reviewed summary of all that information available for you too. So you can use that for citations or client reports or whatever you need. So we'll put that QR code up. But this is so much fun for us, and we're really excited for this Ask a Scientist series. I hope you guys love it as much as we're enthused to do it with you. And we would like for you to continue, join the MI webinar series. Please watch. These topics are going to change and be various all throughout the year. So you're going to get whatever your interest is. Hopefully we'll have something related. And if we don't, send us a question about it. So we'll record a video for you. Reach out to us at info@microbe.com with any questions that you have, anything that you would like to talk about more, or if you just want to set up a call with us so we can discuss things further. I think Sam will agree with me. The best part of our job is sharing what we've learned. And Sam and I have both been doing this for a long time now.
Yeah.
So we have a lot of experience. Yeah, maybe I should not brag about that. But it has been a wonderful journey, and we've learned so much information, and we've worked with some truly amazing people. So we want to share that information with you and to share our enthusiasm with you. And so we hope you'll reach out to us, you'll join us on this series, and you'll help us make 2026 the best year ever. So look forward to hearing from you.
Thank you all for joining.